We'll see some question also, but main focus of today's class is over the glycosidic bond, okay? Because there was one question asked by the student that sugar is also present in the DNA. Is they are capable of forming any kind of glycosidic bond or not? So, can we start with this discussion? Are people comfortable? Kindly unmute yours.
Yes, sir.
Dhanusri?
Yes, sir. Yes, sir.
Okay. So, am I audible properly?
You're audible, sir.
Okay. So, can we start with this question? If there is a sugar is present in DNA and RNA, so what
Okay,
kind of glycosidic bond is possible there? Because you people are in the senior wing, so I think discussion should be there. Properly discussion can make you learn effortlessly. Because I will give you a lots of data and structure, and then at the end you are getting nothing. But if you're starting with the question, then you can give your inputs, and then your brain will involve yourself. Because two separate things are there, your body and brain. If you are learning and your body and brain are behind the learning, then you can grasp a lot. So in the virtual classes, this does not mean that you will sleep. You will learn virtually. No. You have to learn actively. And you have to think, because glycosidic bond everyone knows. If there are two sugars, there will be glycosidic bonds. And also we are going to discuss about the O and N glycosidic bond. And then we are going to discuss most important type of sugars, derivatives like sialic acid, and their role in immunity, virus infection, and many more discussion will be there. So, any idea about the glycosidic bond, in the possibility of glycosidic bond in the DNA and RNA? Yes or no? Just tell me in 2 words.
Sir, this is my first class today, so I think you can just teach, sir. So for the next class, I'll be able to answer properly.
No, I have not taught it before, right? Okay. So we are starting with the this kind of glycosidic bond formation. Earlier, we are going to give you a recap. You can see that. So how to name the DNL sugar? So DNL sugar is named by the orientation, right? So I taught that only. Right, you can see from this ballistic model or kind of Fisher projection formula, a perspective formula, the orientation of hydroxyl group toward the right or left. This is the right hand, okay? So you are going to name as a D-glyceraldehyde, right? And L, when this is a anti-clockwise, you can say. Clockwise, anti-clockwise, you can learn better. Right? So this is clear, I think, how to name the L and D, because these nomenclature are very, very important. So what is the effect of glucose oxidase? This was asked in the CSR examination over the glucose. What product may be possible? So if you are adding glucose oxidase over the D-glucose, because we are writing on glucose, but it does mean always it's a beta D glucose, okay? So always you have to mean that. If I'm asking you about the glucose, so always it will be D-glucose, right? So whenever the D-glucose is reacted with the glucose oxidase, always there will be formation of lactone. D-glucono delta lactone. Is it clear? What will be the product? The product will be lactone. Okay. Lactone, yes. And there will be release of H2O2. So this was asked in the CSR examination. Simply, very simple question was there, but answer many of students were not able to answer properly. Even if you are reacting with the linear form of glucose with some reducing agent, then also there is a possibility of formation of gluconate. Is this clear? Any doubt?
Got it, sir.
Okay, it's a very simple. Already we have discussed. So just it's a recap. How to name the sugar? How to name the alpha or beta? Any idea? Jayaprakash, how to name the alpha and beta sugars?
Yes, sir. Yes,
जयप्रकाश आपकी आवाज कट रही है, तो आप कहीं ऐसी जगह बैठो जहां पे थोड़ा सा हवा आ रही हो, खिड़की या दरवाजे के पास, ठीक है ना बेटा? या छत पे चले जाओ, या कहीं थोड़ा सा ऐसी जगह बैठो जहां पे आपको अच्छा सिग्नल रिसीव हो। ओके, आपकी आवाज बहुत ही कट रही है, बहुत बहुत कट रही है आवाज। चलो, लेट मी एक्सप्लेन। अभी मैं म्यूट कर रहा हूँ आपको, ठीक है? चलो, आपकी आवाज बहुत ही डिस्टर्ब हो रही है। हाँ, राइट। So alpha means if you're writing the sugar like this, then it depends on the position of OH group. And this is the sugar, okay? This is the oxygen, alright? So what you can see here, if the OH is above the plane of the ring, this is the plane of the ring. So above plane of the ring, what will be, you are writing it as a beta, okay? So if this sugar is having OH like this, then this position will be, tell me, alpha. Why alpha? Because below the plane of the ring. Is it clear? Any doubt? So you got the idea, no? So always we are naming starting from here. So for example, you can see here in the sucrose, the name of sucrose will be alpha D glucose, since pyran. This is the pyrene. 6 carbon ring is called as the pyran, and 5 carbon ring will be called as furan. Clear? Any doubt? Any doubt, beta?
Again,
Is this
sir.
clear? Is this clear? What I am discussing?
So can you repeat, sir?
What I am saying ki during the naming of sugar, always you have to name the ring also. No? You have to involve the ring also. So this is a 6 carbon, right? Hexose sugar you can say, or 6 carbon sugar, 6 carbon. So always 6 carbon sugar is named as pyra. And 5 carbon sugar like this, deoxyribose, ribose, or this kind of sugar will be called as furan. Correct? So sucrose is made up of glucose plus fructose. Okay? So glucose is having the 6 carbon, while fructose is having some kind of 5 carbon. You can see nomenclature starts from here. 1, 2, 3, 4, 5. Though it's a 6 carbon, but ultimately the ring is made up of 5 carbon. Okay? So less than 5 carbon you can say, but 5 carbon, this penta structure is called as the furan. This nomenclature start from here: 1, 2, 3, 4, 5 and 6. So this is called as the 6 carbon structure. So such kind of ring will be called as what will be the name of this ring? Tell me. This will be furan, and such type of ring will be called as pyran. Is it clear? बोलो बेटा।
Yes, sir. Yes, sir.
ठीक है ना?
Yes, sir.
So always you should not confuse. This is a pyran or furan? Tell me. This sugar. This sugar is ग्लूकोज इज, फॉरगेट, नोट नहीं बना रहे हो,
Furan. Furan.
फ्यूरॉन. ओके, तो ये याद रखना है आपको, राइट. दिस इज अ फ्यूरॉन. गलत है. फ्यूरॉन किसको बोला था हमने? 5 रिंग को. 6 कार्बन वाले को क्या बोलेंगे? पायरान, राइट. पायरान. ग्लूकोज में क्या है? पायरान है कि फ्यूरॉन है? चलिए बताओ, पायरान है या फ्यूरॉन है? पायरान और फ्यूरॉन. व्हाट काइंड ऑफ शुगर इज दिस? ये है पायरान, फ्यूरॉन नहीं है. क्लियर? डीऑक्सीराइबोस शुगर के एग्जांपल ले लो. दिस इज पायरान और फ्यूरॉन. बताओ. दिस इज फ्यूरॉन. इज इट ओके? ठीक है ये?
Yes, sir. Yes, sir. If five is the furan and 6 is the
हाँ.
pyron.
यस.
Yes.
ठीक है. 5 इज द फ्यूरॉन एंड पी जहां लग जाए वो 6 कार्बन. सो दिस इज अल्फा और बीटा. वो ये चीज above the plane of the ring. दिस इज बीटा.
Beta.
ठीक है ना? अभी नाम समझ आ गया ना, तो इजी हो गया. नाउ
Yes, sir.
इसका नाम मैं पढ़ देता हूं, देन इट विल बी इजी फॉर यू. दिस इज अल्फा डी. दिस इज अल्फा डी, राइट. एंड देन ग्लूकोज का है एंड द रिंग इज पायरॉन. अल्फा डी ग्लूकोज पायरोनेसिल. ये भी पूछा हुआ है एग्जाम में, कई बार एग्जाम में पूछा हुआ है. बीटा डी फ्रक्टोफ्यूरनोसाइड. तो ये फ्रक्टोज का है, फ्यूरॉन रिंग है. दैट्स व्हाई दिस इज कॉल्ड एज फ्रक्टोफ्यूरनोसाइड्स. तो कई बार आपको शॉर्टकट में नोमेनक्लेचर मिलता है, राइट. सो यू शुड नॉट कंफ्यूज. ग्लूको के लिए द वर्ड इज GLC. आई हैव गिवन द एब्रिविएशन ऑलरेडी. फ्रक्टोज के लिए FRU, फ्यूकोज के लिए FUC, राइट. गैलेक्टोज के लिए गैल, ग्लूकोज के लिए GLC, मैनोज के लिए मैन, रेमनोज के लिए RH, राइबोज के लिए रिब, जाइलोज के लिए जाइल. ठीक है, याद रहेगा. एरिबनोज के लिए आरा.
Yes, sir. Yes, sir.
ग्लूकोर्निक एसिड GLCA.
Yes, CA.
ठीक है ना? ओके. गैलेक्टोसामिन गैलिन.
GAIN.
हाँ, GLCA.
GLCN.
हाँ, अमीन लग गया तो आइए. ग्लूकोर्निक एसिड A, GLCA. ठीक है ना? एनेस्टल ग्लूकोसामिन नाइट, गैल नाइट. एनेस्टल ग्लूकोसामिन, एसिटल ग्लूकोसामिन, एनेस्टल ग्लूकोसामिन, गैलेक्टोसामिन ये है. तो गैल नाइट, गैलेक्टोसा, गैल नाइट. ठीक है. एंड एसिटल ग्लूकोसामिन GLC NAC. तो यहाँ पे ये लिया है ग्लूकोसामिन, ग्लूकोज को. यहाँ गैलेक्टोज के लिए, गैलेक्टोज के लिए क्या है? गैल. यहाँ पे चेक करो, गैलेक्टोज के लिए क्या है? गैल. ग्लूकोज के लिए GLC. ठीक है ना? समझ आ रही बात? बी इंटरेक्टिव है ना, डिस्कस करते रहो, हाँ ना बोलते रहो, कोई शर्म की बात नहीं पढ़ाई, राइट. इफ यू हैव एनी प्रॉब्लम काइंडली से विदाउट एनी हेजिटेशन.
Yes, sir.
हाइड्रोजनिक एसिड. हाँ, ठीक है ना. तो यू आर गेटिंग ना मिरामिक एसिड म्यूर. तो ये सारे एब्रिविएशन मिलेंगे when I will be dealing with the peptide bond of bacterial structure, or I will be dealing with the membrane structure, viral structure. They are interacting with the human membrane. तो ये क्वेश्चन काफी आता है रिसर्च में कि how the virus is recognizing the or interacting or entering the mammalian membrane. है ना? Virus is interacting with the mammalian membrane. How? Or if RBC is there, then also WBC is there, correct? Right? So WBC, RBC, blood type. Okay? All blood type is going to differ in what? Sugar type. Okay? Sugar type. So one question frequently you have been asked in the examination that suppose 4 bases हैं DNA में A, G, T, C. So how many combination is possible? ठीक है. इसी तरह से पेप्टाइड में क्वेश्चन आता है या प्रोटीन में. If there is a 20 amino acid, how many polypeptide possible? आता है ना इस तरह क्वेश्चन. How many polypeptide?
Yes, sir.
तो इसका आंसर कैसे देते हो और इसका आंसर कैसे देते हो? तो 3 के कोडॉन में 3 का कॉम्बिनेशन होता है. ठीक है. तो 4 की पावर होता है 3, राइट? तो 4 into 4 into 4 कितना हो जाता है? 64. इसमें नॉनसेंस कोडॉन 3 होते हैं तो आपका टोटल कॉम्बिनेशन हो जाता है 60. पेप्टाइड में कैसे करते हो? 20 के पावर 2 के पावर 20 because 2 amino acids are going to form 1 peptide bond. 2 के power 20, what will be the structure, what will be the combination or result? That will be prediction of how many polypeptide bond is possible. ठीक है. अभी मेरा एक वेबिनार होने जा रहा है 2 नंबर, देखा था पोस्टर, नेक्स्ट जनरेशन सीक्वेंसिंग पे. काफी एक्साइटिंग है. बहुत बड़ी कंपनी है नोवाजेन, राइट? So they have invited me to give that lecture. Are you interested in that? बिल्कुल इंटरेस्ट खत्म हो गया है. हैं?
Interested.
है ना? वो 1 घंटे का होगा. That you can attend. नेटवर्क वगैरह थोड़ा सही रखना. पैक वगैरह अपना चेक कर लो. अभी तुम्हारा नेटवर्क सही आ रहा है. पहले पता नहीं क्यों कट रहा था बहुत. मेरे ख्याल से तुम्हारा नेटवर्क दूसरा था वो, है ना?
सर वो मोबाइल से था, अभी लैपटॉप से है तो फिर.
हाँ सही है. ठीक है ना. तो ये सारा हमने डिस्कस कर लिया था. ट्रायोजेज, ग्लिसरॉल्डिहाइड, डाइहैड्रोक्सिस्टोन, टेट्रोजेज, इरेथ्रोज, है ना, पेंटोजेज, राइबोलोज, साइलोज, हेक्सोजेज. दीपावली की छुट्टी भी कर रहे हैं मंडे से लेके ट्यूसडे तक. मंडे ट्यूसडे 2 दिन की रहेगी. ठीक है. मंडे ट्यूसडे वेडनेसडे 3 दिन रहेगी. इसके बाद चलो आज हम जो पढ़ने वाले हैं ये सारा हो चुका था. माल्टोज, 2 ग्लूकोज मॉलिक्यूल, लैक्टोज, ग्लूकोज, गैलेक्टोज, सुक्रोज, केन शुगर, ग्लूकोज, फ्रक्टोज, है ना. सो कार्बोहाइड्रेट ऑलरेडी वी हैव डिस्कस्ड, मेड अप ऑफ एल्डोज एंड कीटोज. तो यहाँ कायरल सेंटर की बात करी थी हमने, राइट? कि अगर एक कायरल सेंटर है तो हाउ मेनी स्टीरियोआइसोमर पॉसिबल? 2. इफ 4 chiral center are there, how many combination are possible or stereoisomer possible? 16. All right. So already we have discussed this one. ठीक है. So already we have discussed about the epimers. What are the epimers and how they are different? So epimers are always occur between the 2 molecules which are having the same molecular formula.
Same molecules.
They are different in the single carbon atom, somewhere in the orientation of hydroxy group. Okay. So if you are comparing the D-mannose and D-glucose, they are epimer. Epimer means different at carbon 2. Okay. So if you are comparing the D-glucose with the D-galactose, they are epimer at carbon number 4. So this you must remember properly. Okay. So pyranose concept just we have discussed. There are 5 carbon sugars like ribose, deoxyribose, 3 carbon sugar, glyceraldehyde, 6 carbon sugar, glucose, fructose, galactose. Okay. And there are sugar family. Do you know about the sugar family? There are large number of sugar family called the amino sugars like galactosamine, manosamine, right? Deoxy sugars,
Yes.
fructose, rannose, right?
Yes, sir.
And in the
Randolph.
glucose family, you can see glucose, already we have discussed. Then its derivative are glucosamine and N-acetyl glucosamine, NAG, and NAM. They are the part of bacterial peptide. So NAM and NAG frequently asked. Sometimes they can ask you this structure. What is this? And then muramic acid. This is also the part of bacterial cell wall. Okay. So that's why I think more than protein, sugar family is important, because many of the teachers going to ignore the sugar families in detailed discussion. Right. So, but I decided that I should discuss in very depth. What is the acidic sugar? Sialic acid. Sialic acid is a very good example of acidic sugar. Why this
Sugar.
is acidic sugar? Because you can see they are having the carboxyl group. Okay.
Group.
So they are called as the acidic sugar. Sialic acid. Sialic acid is very important in point of various functional role in human physiology, like cell to cell interaction. How the cell to cell is interacting? They are interacting because of presence of sialic acid. If sialic acid is not present, so probably the cell to cell interaction will not be there, properly will not be there. Right? So communication, for the communication purpose with the different cells, cell signaling, and even carbohydrate protein interaction and cellular aggregation. What is important? Sialic acid. Okay? Sialic acid. You are studying a lot of developmental processes in the unit 5 of CSIR syllabus. Okay? Immune reactions in the unit 4. All they are because of sialic acid. If sialic acid is absent, everything is absent. Okay? No developmental process, no cellular aggregation, no cell signaling. Okay? Reproduction, neurobiology means human brain having the largest number of sialic acid, and that's why you are genius or dumb. Okay? If you are having a very good number of sialic acid in your brain, you are genius. Your capturing power is good. You are motivated to learn. If you are dumb, means you have very less amount of sialic acid in the brain. जैसे हमारे ग्रुप में देखो बहुत सारे बच्चे हैं, ठीक है, but all they have very low sialic acid in the brain. Why? Because उनका ब्रेन ही नहीं काम करता कि if you are inviting, है ना, आपको क्लास करना है, ग्रुप में है वो, but they are brain dead because they cannot think of beyond that. है ना? So your brain is genius because you are receptive, you are interacting, you are searching, you are solving a problem, है ना? Then you are making the progress. Your life is get stuck. As soon as you take the decision, no. If you take the decision yes, your life is going to proceed. Okay? धनुश्री तो क्लास ने अवॉइड कर दिया था, क्लास नहीं करना, तो मैंने कहा चलो बुलाओ है ना. She wants to learn but has some problem. चलो interaction is very good. Sialic acid के example दे रहा हूँ in the term of immune response. So suppose virus attack is there, then pathogen attack is there, complement activation is there. All these processes is because of presence of sialic acid. Why? Because sialic acid can modify the immune system. Sialic acid can modify the immune system. So what is the benefit of modifying the immune system? You can enhance or you can slow down the defense activity, right? So for example, if you are under attack of influenza virus, okay, so if you are under attack of influenza virus, then the largest number of immunoglobulin secreted will be IgA. Which immunoglobulin will be secreted? Tell me. IgA. But IgA will not be able to defend if there is an absence of sialic acid. Dhanusri, are you comfortable? in Hindi?
Yes.
Because I don't know. Hmm?
Yes, sir. I can understand Hindi.
Okay, you are from Kerala? From where?
No, sir. Karnataka.
Acha, Karnataka, right. Very good. So, you got any college after CUET entrance or still waiting for?
No, sir. Not yet. I'm still waiting, sir. I have applied. The merit lists have not been out yet.
Okay, okay. So, your preference is which university?
University of Hyderabad, sir. Hyderabad and Kerala.
Kerala, okay. Very good. They are very good university. Chalo, so they have a very good, I think you will select biotechnology stream or some other.
No, sir. I'll select biotechnology.
That's I know. Because I myself belong to biotechnology. This is the most advanced science. This has been ignored by our Prime Minister Modi sir. But no worry. This is the most advanced and it will be the most advanced science because you will see the Bangalore, Karnataka, all they are flourishing because of biotechnology. Okay, Pune, all they are flourishing. If you want to make a state or country flourishing, then biotechnology is must.
Yes.
Biotechnology is very advanced because huge amount of brain will be required to think and to act, to learn. And I am happy that you are learning at this stage, very advanced science.
Yes,
we are discussing at very advanced stage. Okay. So we are going to discuss all these things in very deep.
I'm making the points, sir. So I'll learn this again.
I know, because you are not aware of so many things. So it's my duty to ask so that if any problem is there, your hesitation must be removed. You should feel interactive because, we are talking brain to brain. We are not learning with the PPT or virtual mode. We are talking brain to brain and for brain to brain interaction, you must feel comfortable and familiar with the teacher. You have a good connect with the teacher. For example, Jayaprakash is very much connected to me. और फोन करता है कई बार पूछता है सर ये क्या है वैसे क्या है मतलब ही इज वेरी इंटरेस्टेड इन लर्निंग तो एक टीचर बहुत हैप्पी होता है कि व्हेन देयर स्टूडेंट वांट्स टू लर्न टीचर को क्या है मैं तो ऐसे पढ़ा रहूंगा आप मत पूछो कुछ मैं आराम से पढ़ा के निकल जाना है राइट सो माय एफर्ट इज टू मेक यू aware of the latest thing also, latest discovery. For example, I prepared this slide beyond your expectation because it's a part of the things and it must be covered, but most of the teachers are not covering this. What is the role of sialic acid in platelets? So I am discussing here because you must aware that this is a very important carbohydrate and they are activating and playing so much roles. If your Zoom is disconnected, then please connect after 10 minutes, okay? Again. We will be discussing continuously and from tomorrow I think I will be on the OBS so I am just trying to familiar with the OBS because I am not familiar with that system. Then we'll see. So sialic acid of human platelets has the N-acetyl acid. We just discussed in abbreviation part. We have discussed together. So then whatever the platelets are going to play a role like aggregation, adhesion, all this is because of sialic acid. Clear? So what is the next? Brain is the organ which already we have discussed. Highest level of sialic acid in the body, much of it in the form of gangliosides. Does influence of sialic acid? Yes, sialic acid linked to glycoprotein and ganglioside is used by many viruses as a receptor for cell entry. This is the report of the article 20 July 2013. Okay, these viruses include important human and animal pathogens such as influenza, parainfluenza, even COVID, mumps, corona, noro, rota, DNA tumor viruses. Got the point? So latest research is mostly based on the sialic acid, understanding of sialic acid interaction. क्योंकि अगर साइलिक एसिड है तो वायरस इंटरेक्ट करके इन्फेक्ट कर रहा है अगर साइलिक एसिड एब्सेंट कर दे तब भी प्रॉब्लम है ना कि अगर आपने साइलिक एसिड एब्सेंट कर दिया तो व्हाट विल बी द फ्यूचर कुछ भी नहीं होगा क्योंकि ब्रेन काम नहीं करेगा फिर प्लेटलेट्स एग्रीगेट नहीं करेंगे डिफेंस जिसे हमने बताया एंटी इम्यूनोग्लोबुलिन आईजीए they are very specifically interfering with the cell surface attachment of influenza virus. तो वो भी है कि वायरस को भी हेल्प कर रहा है और हमारे बॉडी डिफेंस को भी हेल्प कर रहा है. तो दिस हाउ टू सॉल्व दिस थिंग्स, मे बी इन फ्यूचर सम साइंटिस्ट या आप लोग कुछ कर सको. So my duty is to make aware of all these things. Okay. So we will discuss now about the glycosidic bond. Have you heard about the glycosidic bond?
Yes, sir.
What is this bond?
The joining of monosaccharide units.
Joining of two monosaccharide units. For example, alpha D glucose, beta D glucose. They are combining after the condensation and forming this maltose structure, and in between these two monosaccharides are bond called as the glycosidic bond, often represented by symbol like this. Okay, so this is called as the O glycosidic bond. O glycosidic bond. Okay. And there is another type of glycosidic bond, that is N glycosidic bond. So what is the difference between two? When there is a N carbon atom, right? You can see here. This is N link structure. Then it's called as N linked glycosidic bond. This is O linked because nitrogen is not there, only hydrogen is there. So H linked with O, that's why O linked. This is NH linked with O, so that's why this is N linked. Serine is mostly interacting to form O linked. Asparagine is mostly interacting asparagine. Asparagine. I don't know how to write. And serine. Okay, this is the serine. Serine is what? Amino acid, right? This is amino acid. Okay, and this is sugar. Sugar to sugar also glycosidic bond, but if amino acid, what you can write? If amino acid plus sugar, then there are two type of bonds. O-linked glycosidic and N-linked glycosidic bonds. ठीक है? For example, you can see the representation of O-linked bond like this. Okay. And N-linked bond. How to represent N-linked bond? This is the SARD form, red color, fucose. Okay, this is the fucose. This is red color, galactosamine. Okay. This is mannose, this is galactose. Every symbol has been there. So that's why I was making you familiar for the symbols. Right, Peter? Now the next point. N-linked glycoproteins. Glycoproteins are the best example of N-linked sugar. You can see the two terms, glyco and proteins. What do you mean by the glycoproteins? Any idea? What do you mean by glycoproteins? Glyco means
Glucose.
Sugar.
Per molecule.
Sugar when interacting with the proteins. Then this is called as the glycoproteins. Have you heard about the glycoproteins?
Yes, sir.
What is the role played by the glycoproteins? As a receptors. Mostly receptors are made up of glycoprotein.
Okay.
Okay, so whenever your body is going to identify between the foreign molecule and our own molecule, this sugar is kind of acting as a marker. This is very advanced thing. Have you heard about the marker? And if you are acting in the laboratory, if you are working in the laboratory, then your supervisor might talk about these markers, receptors. Okay. So mostly this receptor is isolated and identified. So in this receptor, you might be knowing there is a membrane structure. Membrane structure is made up of, tell me. Membrane structure is made up of
Hydrophobic head and hydrophilic tail.
exactly what it's called as
Lipid bilayer.
bilayer. Yes, phospholipid bilayer. So you can see there are two types of proteins: intrinsic proteins and extrinsic proteins. Okay, right? This is extrinsic proteins, and they are which protein? Extrinsic and intrinsic protein. So they have the sugar molecule, and the sugar molecule act as receptor. Clear? So this is the role of aniline glycoprotein. So what about the erythrocyte? Erythrocyte. Have you heard about the erythrocyte? Erythrocyte have the
RBC, sir.
surface protein called as the glycophorin A. So erythrocyte membrane have outer surface called as the